Okay, let's now take a look at water flow in the small and large intestine. Water flow is an important factor because enough water needs to be into the intestine in order to make sure that the proper digestive events occur. Remember that the digestion of our food sources is largely hydrolysis, which is in need of quite a bit of water, and then also water is important for solubilization, and it's also important for material movement through the intestines. Now, where does this water come from? Well, we have approximately 9 liters of water moving through our intestine on a daily basis, and it's estimated approximately 1 to 2 liters comes from food and drink. That means the rest of it must come from other internal sources. So what we have to consider is that the small intestine will put out approximately 1.5 liters of water per day. Now, how does water get secreted from the inside of the body into the lumen of the intestine? Now, if we look at our cell, our small intestine epithelial cell, we have to consider the basal surface and the apical surface because obviously the process of water delivery into the intestine needs to be a directional process. So if we have the lumen of the intestine up here, this of course is the apical surface, this is the basal surface. The movement of water is going to follow ions, and so therefore we are not going to be moving water or pumping water. We are going to be moving ions, and then of course by the process of osmosis water will follow the ions. Now in the basal surface here we have our classical sodium potassium ATPase transporter. So remember, this is primary active transport. We're going to hydrolyze ATP and use this energy to drive this pump. So essentially we're going to be pushing sodium out of the cell into the interstitial space. Now in order to make sure that we don't have any depletion of potassium, because potassium's concentration obviously is much lower than sodium, we are going to have a potassium channel that is going to allow the gradient to dissipate of potassium. This channel is just a simple potassium ion channel, and then it can feed the sodium potassium ATPase. So in this manner we are able to get the sodium from the inside of the cell to the outside. Now once we have the sodium on the outside in the interstitial space, we have another transporter that is actually a tripartite transporter. This transporter is going to transport sodium into the cell. It's also going to transport potassium into the cell. And for our purposes, most importantly, we are going to transport chloride ion into the cell. So essentially the gradient of sodium we're establishing with the sodium potassium ATPase, this sodium is going to allow the driving of this tripartite pump. The importance of getting chloride ion into the cell comes when we look at the chloride channel. Now this is the CFTR, and remember CFTR stands for cystic fibrosis transport regulator. This is a chloride channel. The importance of this chloride channel is it is a regulated chloride channel. So what is going to control this process is a second messenger, cyclic AMP, which is going to activate the CFTR, which means open the channel to allow chloride to flow through the chloride channel into the lumen. Now, what's important about this is that we are going to start having a electrochemical gradient of chloride ion, and so naturally physics will take over and sodium will transport from the interstitial space through the cellular junctions. So this would be an adjoining cell. And the chloride will flow in between in the intercellular space in order to neutralize the chloride ion. Now that we have our salts moving into the lumen, water will flow from the interstitial space into the lumen of the intestine. Now, another key element to keep in mind is that you only want this under regulated conditions. So what exactly is going to regulate this and control it? Well, notice we have cyclic AMP here, which is the second messenger. So that means we need to have the activation of adenylate cyclase. And recall that adenylate cyclase is typically located near the plasma membrane surface, and the function of adenylate cyclase is to generate cyclic AMP from ATP. Now recall, back in your knowledge of signal transduction events, what activates cyclic AMP. That will be a heterotrimeric G protein. Now if we back up one step further, what activates the heterotrimeric G protein, which is essentially the G alpha subunit, and that is going to be a 7-transmembrane receptor, and this receptor binds a small effector called VIP for vasoactive intestinal peptide. Now, the vasointestinal peptide, this is going to activate the heterotrimeric G protein, which in turn will activate adenyl cyclase, which in turn will increase the concentration of cytosolic cyclic AMP, thereby by opening the chloride channel for this process to occur. Where does vasoactive intestinal peptide come from? Enteroendocrine cells in the small intestine. So these enteroendocrine cells are activated by chyme. Now recall, chyme is a mixture of digestive juices, juices from the stomach, from our dietary sources, juices from our pancreas, digestion products, bile. All of this here will activate the enteroendocrine cell to secrete vasoactive intestinal peptide. Makes sense, in order to increase the flow of water into the lumen to aid digestion. The other thing that vasoactive intestinal peptide does is it stimulates buffer secretion. This is in the pancreas. Recall that the pancreas is going to be secreting bicarb to neutralize the stomach acid, and VIP also helps this Now, the main hormone that is doing this is secretin. We'll cover this a little bit later, but vasointestinal peptide also does this. The peptide also inhibits gastric secretions. And this makes sense because once the chyme is now down into the small intestine, we can begin to shut down gastric secretion, that being primarily acid production, because now the food is moving down the digestive system. The final thing that VIP does is it dilates intestinal capillaries. Again, this makes logical sense because now we have the digestive material in the lumen of the intestine. It's being processed and absorbed into the body. And so let's dilate the capillaries so that we have sufficient flow of nutrients from the intestine to the rest of the body. Now, once we have water secreted into the lumen of the intestine by the small intestine, we have our 1.5 liters. Eventually, we have to start retracting this water. We have to absorb this water from the lumen. Otherwise, we're going to have a situation of runny diarrhea. We actually only end up losing about 100 milliliters of water in the feces. Now, compare that with 9 liters of water being put into the intestine. So there's a significant water absorption property. So how is water absorbed in the small intestine? Let's look at our small intestinal epithelial cell again. And again, the movement of water is not going to be done by any pumps. It is going to be following a gradient of ions and other osmotically active molecules. So on the surface, we are going to have a transporter. This transporter is called SGLT1, which stands for sodium, glucose, transporter, type 1. And this transport is a symporter, which means that we are going to be able to transport sodium from the lumen as well as glucose. So being a symporter, both ions need to be bound to the transporter before the transporter can shuttle these molecules in. If sodium is bound, or if glucose is bound alone, the transporter won't work. So we need them both together. On the basal surface, we have, once again, our familiar sodium potassium ATPase. Now this is using, again, the energy of ATP hydrolysis to drive this sodium out. So we have a very large flow of sodium into the body. We also are establishing a gradient of sodium, which is driving the glucose, and that is causing a glucose gradient to form. And on the transporter, on the basal surface, this is called GLUT2 for the glucose transporter, type 2. This transporter is able to respond to the chemical gradient of glucose that's in the cell created by SGLT1 and transport glucose. Now, we're going to see a little bit later when we talk about digestion of carbohydrates that this is the primary mechanism of getting glucose from the lumen of the intestine into the body, into the blood, which is down here. This is also the primary mechanism of getting water in, because notice these ions plus osmotically active molecules, such as glucose, as these are flowing into the body, water will flow in with it, allowing us to absorb the water from the small intestine lumen. Now in the next video, we'll take a look at water reabsorption from the large intestine, because this will be our last chance to capture the water inside the body.